Good morning, friends. I am Siddharth Kalika. I teach you biology, as you all know. I am starting from progressive coaching classes. As we have all know that the coronavirus has been impacting in our social and economic life as well as your study life as well. So being a second year student, you should have focus on your study. So we are starting our second year syllabus from the DNA packaging. That is a part of your genetics syllabus. Right from the beginning, if we have to deal with these things, you must have to know this some basic terms before going to the main things. So the first thing, you must have to know the difference between the DNA and the gene. So we all know that the DNA is our genetic material, right? But some students mix the term DNA with the gene. So if we suppose imagine this is our DNA, though it's a helical structure, I am just simplifying the structure in a parallel fashion. So the functional part of the gene, the functional part of the DNA that transcribes to a mRNA and finally gives me a functional protein is my gene. That means all the part of the DNA is not the gene. The functional part that is going to give me the protein is my gene. Then, if we analyze the cell, within the cell we have the nucleus, that is like the brain of our body, that contains the genetic material inside. When the cell is at rest, means that the cell is not preparing for any cell division. Then the genetic material is in a thread-like haphazardly arranged inside the nucleus, and that we call as chromatin. But when these materials arrange themselves just before cell division occurs, we call that structure as the chromosome. Again, one very basic point. If someone asks you the structure of a chromosome, always imagine a structure like this, but not this. Why so? Because this is the chromosome in metaphage, not this is actually the structure of chromosome. I think you have studied this thing in the cell cycle chapter of your first year syllabus. So the same thing, that is my genetic material, that is my chromatin, arranges to give the appearance of a chromosome, and this is what we are going to study. We are going to study in this headline, that is our DNA packaging. And when the chromosome appears this shape in the metaphage, we call this individual structures as chromatin. So don't make the mistake between chromatin and chromatid. So before dealing with the subject, that is DNA packaging, you must understand why we are studying this. The point is that if you imagine the double helical structure of DNA, we know that the building molecules, the building blocks of a DNA molecule will be my deoxyribonucleotide or simply nucleotides. The number of nucleotides in one single cell is almost 3.2 billion. So the point is that this is a cell. We cannot see a cell with our naked eye. Inside this we have the nucleus. We can imagine of viewing this with our naked eye. And inside this nucleus we have this much number of nucleotides arranged in a structure of a DNA. So how they are being arranged in a very sophisticated manner, in a very well-fashioned manner, to to have accommodation of this structure inside this. This is what we are going to start. The point is that suppose you are going to a vacation in any places, suppose you are going to vacation in Goa, what we will think like is that as much as new clothes you have or as much as variety of clothes you will have in your trip, you will be more fashionable for you. So what will you try to do? You will always try to have a very planned packing of your clothes. That is what the DNA is doing for itself. It is packing its nucleotides in a very specialized fashion so that it can accommodate maximum number of nucleotides in a very small space. So if you just imagine the DNA, its diameter is 2 nanometer. Its diameter is 2 nanometer. So what it does inside the nucleus, what it does, this DNA stretch or DNA molecule rotates around the histone proteins. So what are histone proteins? These are basically some of the proteins, basic proteins present inside the nucleus. So basically we are having few different types of histones like H2A, H2B, H3 and H4. So you imagine there are 8 coscoballs, 4 coscoballs at the front and 4 coscoballs at the back. So this makes the histone core around which this DNA molecules rotates in a fashion. So I am drawing one single stretch, but you imagine that one single stretch is carrying both these 2 strands or the DNA molecule. So it rotates like this. It's one complete turn followed by the left. So we call this turn as 1 3/4 turn. Not 2 complete turn, 1 complete turn and 3/4 turn. And from this point to this point, as you imagine, this is my DNA stretch. It is having some nucleotides. The number of nucleotides from this position to this position is 146 to 147 in different conditions. And at the opening of the structure, we have another histone that is we call as H1. So this whole structure, what we called as nucleosome, and this is called as histone octamer. Octa means 8. Why 8? 1, 2, 3, 4 at the front, and same H2A once, H2B another again, H3 once more, and H4 once more at the back. That is why histones 8 times, making it histone octamer. So this whole structure with your histones at this one along with H1 and at the end stretch, we call this structure as nucleosome. So what it happens, it makes a structure like this, and this is we call as beads on a string structure. So this attains this structure where you can imagine these are the beads. So that you can imagine a thread where you are putting some rice-like structure, and this rice-like structure are my beads. And actually these are nothing but this nucleosomes like this. So when it attains this structure, it increases the diameter to about 10 nanometer. What it can do, in 2 nanometer, it just accommodated this much number of nucleotides. But now you imagine at this position, if you just imagine one position at each stretch, at this position you will having 4 nucleotides at least. So the number are increasing in a very limited space. What happens next? We call this as a solenoid structure. So what's a solenoid? It's a kind of structure like this, So imagine this stretch of DNA molecule along with the histones in anywhere in the solenoid. Here it can be here or here. So what happens? The diameter becomes 30 nanometer, but in one single stretch, you can imagine how many more nucleotides are being associated, because as you imagine this single line represents this structure. So at this position, you can imagine any position, we will having many nucleotides in a single pact. This modifies to another complex structure. I have drawn the upper structures. So taking non-histone protein as a base, remember this was histone, this base is non-histone. This is also called as scaffold protein. So so imagine this stretch of solenoids, one after another, millions of solenoids. You imagine this in this structure. So in this stretch or anywhere, you will have this stretch of solenoids when the diameter becomes 300 nanometer, but in one single point, you can imagine many nucleotides compared to the previous structure, which then gets packed more and more when the cell actually enters in the prophase stage that we studied in our first year syllabus. That this non-histone protein makes a stretch to give you an appearance of the chromosome. So this is basically the non-histone protein or the scaffold protein, where you can imagine these structures are present like this, but it is not needed to draw like this, where the diameter becomes 700 nanometer, which is followed by the structure you imagine earlier to be chromosome, but actually the chromosome in metaphase, where the chromatin molecules get split to give you the individual chromatid. So these are individually my chromatid, and which actually gives me the diameter of 1400 nanometer. So you can relate this thing to your first year knowledge that we studied in cell cycle. So in cell cycle we studied that it is comprising of a phase that is G1, S, G2 and M phase. So we studied that the DNA molecules get doubled in the S phase, but we cannot visualize that the something that double, actually we can visualize in the M phase, and most specifically in the metaphase. So yeah, in this structure itself, the amount of DNA was already being doubled, but we cannot visualize it, and the double amount can be visualized clearly in the metaphase structure, where the diameter gets doubled to 1400 nanometer. So the point is, again, you remember DNA packaging. So what is the need of this packaging? You again have to go back to your first year knowledge that when the cell has to divide, each new cell has to get the same copy from the parent cell. So if this is a cell, this is the nucleus, whatever be the material, if it is the earlier amount that is not being doubled, even if suppose it is being doubled now, you cannot differentiate the two molecules easily. You cannot differentiate which part will be going to this number 1 cell, which part will be going to number 2 cell. For this anomaly, what it does, it arranges themselves according to their chromosome number. And when it arranges, the same amount comes twice in one chromosome, which splits to 2 chromatid in metaphase. That is why this is the chromosome in metaphase. And when it gets splits in the next phase, that is anaphase, suppose it gets splits, it will be acting as a new chromosome for the next cells. So that is the most significant thing about DNA packaging, that it arranges its DNA molecule so that when splitting occurs, whatever there in this position, it should be the same thing in this position, because both these two cells have to get everything equally from the parent cell. Thank you.